Okay, so I think we're just gonna make a start at this point. I realize that everyone has a lot going on with the beginning of the semester, but it's important for me that we have these optional weekly live online meetings. So we had one earlier today. This is the week 2 recording. And we're now in the evening, 8 o'clock, tackling the material from week 2, which focused on the blood types. And since I already have offered one of these meetings today for this week, I don't want to keep repeating the same stuff. It kind of doesn't serve a purpose if you end up watching one of these videos as well, or these videos as well. So rather than focusing on the reviewing the key points of the chapter on blood, what I'm going to focus tonight, I think, unless someone shows up with questions that differ from this, what I'm going to focus on is to look at the lab that you will be completing, or that you actually already completed if you attended in person. So our lab was on Wednesday, and you did a little bit of blood typing, and then you looked at the blood donor recipient matching and how all that works. So the very first thing that I want to do is to share my screen as it is for the seeing the view of our course on the canvas. So again, your view will look slightly different, but it is very closely resembling what you'll see here. And for the week 2 module, I'm actually going to jump to the student view. I think it's going to serve everyone's time the best. So for the week 2 module, which is about the blood, when you scroll to the assignments, you'll see that our lab for today was blood typing and transfusions in class activity. So that's going to be due at the end of this week. So on Sunday, which is January 30th, I believe, at 1 minute before midnight. So you have all the way till the end of the Sunday to tackle that, just as you have all the way to the same deadline to tackle the blood quiz and blood anatomy practice. These labs largely, and especially as we move further onwards, require you to be present in the class to complete elements of those. There will be elements that are impossible for you to complete if you're not in the class. But luckily for anyone who missed it this week, there's a significant amount of this lab that you can also complete even if you didn't attend to the class portion. So you'll just miss out on a little bit. So once you click in there, you'll see a description of the activity. And what you'll need to do, you need to download the blood typing and transfusions worksheet. This is the lab report that you complete, and this is the lab report that you submit to me to get points for completing this lab. And we often do provide you with a grading matrix that breaks it to quite a bit of a detail that how your grade for completing this lab will be calculated. So I really encourage, when you're given a tool that tells you how you're being graded, take that. Have a look of what you are expected to answer and to what level to get points. And remember that these points add up towards your final grade. So you can complete most of the boxes that we have here. So 4 of the first ones, simply as what we call a dry lab. So simply using your knowledge from the lectures and using your notes, using your textbook, you can answer to many of these questions. but you will miss out on the blood typing card and identification if you were not present in the class. So that's just something that you have to be at peace with, that you might lose out on those points. So what we're going to do, we're going to work together through this blood typing and transfusions worksheet. But before we jump in there, I want to take a little bit of time to remind us how the blood typing worked. And for that purpose, I have prepared a nice little PowerPoint for us. Let's have a look of that together. Let me find the right screen to share with you. You should be seeing the screen of mine now. We use this in the class, and it's gonna follow very closely to what you already saw in the class, but I think it's a good way to get us oriented on how to tackle this particular lab. So the lab that we're looking at when we're talking about blood typing, we're gonna be focusing largely on what's going on with the red blood cells and what's going on at the plasma of the blood. And those two things will define whether we are able to give a transfusion from one individual to another or not, based on whether the blood types match or they don't match, or whether it's acceptable even if it's not a match. So let's have a look of that. And I really enjoy talking about blood types because there's so many misconceptions regarding blood. As I've seen some really interesting ones, for example, that you could eat based on your blood type, or that maybe your personality would be impacted by your blood type. And I think that one of the kind of most interesting one, when I ended up to the black hole of exploring what's available online, was that you could date based on your blood. That really left me wondering that there's a lot of confusion in this world about that. And I think that the confusion comes from the historical background that back in the olden ages, you actually needed to get a blood test before you were allowed to marry, to get the marriage certificate and go through the whole process. So a lot of people seem to have from these old days this conception that, okay, so I guess that blood type really determines a lot of things. Well, I'm going to be honest, this needing to get a blood test before you marry had nothing to do with whether you're a compatible couple. It had everything to do with us trying to prevent spreading of sexually transmitted diseases or illnesses or infections. So really, that's what it all was about. One thing that I think that leads into this, that people find blood types confusing, is because there are so many different blood groups. There's actually 32 different kinds of systems of classifying the different blood groups. But really, it's going to be two of those that we're going to be focusing. We're going to spend most of our time talking about ABO blood grouping system, and then we'll spend a little bit time jumping to talk about Rh positivity or Rh negativity. And especially the Rh positive negative will become more obvious once we work through the lab together. What I find adorable is that we actually share our ABO blood type and our blood groups, ABO blood groups, and also Rh groups with our closest evolutionary relatives, which are apes, chimpanzees, and gorillas. And what you can do, you can check actually the donor and recipient match with very many different kinds of tables that you find floating around and made available for you. And sometimes my students are really eager and they go through the trouble of memorizing these. Please don't do that. don't make your life more difficult than it has to be. What you have is me to help you to avoid that. So what we will do, rather than memorizing a table, we'll learn how to understand the table. Before going to that, let's have a look of a little bit of the history. So blood groups were discovered sometime in 1900 by this gentleman called Karl Landsteiner. And what he noticed in his work was that some people were saved due to blood transfusions. Well, some people died when they received blood transfusion. So he was really curious about that and wondering about that. And I think that that's a good question. And actually, that good question won him a Nobel Prize in 1930s. So one thing that we'll notice, and we have to kind of keep at the forefront of our mind, is that blood types are inherited genetic traits. So your mother and father will contribute to what blood type you are. And this makes me think of this whole concept of dating based on your blood again. I find it very peculiar thinking that, okay, maybe that's what's throwing some people off. But other thing that it makes me think of when I think of that is this whole concept, okay, we have inherited traits. One of the many inherited traits that we inherit from our parents is, for example, whether your earlobe is attached or non-attached. So do you think that that would be a good cause for choosing your partner, dating based on earlobes? I don't think that you think so, and I certainly do not. So we're not going to go there. One thing that I think adds up to this confusion about the blood typing is that ABO blood types are not inherited based on the Mendelian inheritance patterns. Instead, it gets actually more complicated because we're going to be looking at 3 different alleles rather than what we've seen a lot of time in Mendelian inheritance patterns where we're looking at 2 alleles. So let's think through what's going on. And to do that, I'm going to draw an analog. So work with me. This is going to be a little bit of stretch, but if you work with me, we'll get there together. So red blood cells look a little bit like donuts. And on top of donuts, what you'll end up finding are these sprinkles. Similarly, on top of red blood cells, you have something that we call as antigens. So you can do in this analog, you can draw an analog between the donut being the red blood cell. I do realize that real red blood cell doesn't have a hole in the middle of it, but it has a little depression. It had that biconcave shape, disc shape. So it's close enough if you have good imagination. And these sprinkles are going to be the antigens on the surface, similar to the antigens that we find on the surface of red blood cells. And the purpose of these antigens is that our body is constantly screening whether something is part of us, so it's part of your own body, and we're happy for having that cell in your body because it's part of you, or whether something is foreign and it might be an invader, some kind of a pathogen, something that threatens your well-being. That's what your immune surveillance is doing all the time, screening for all kinds of potential invaders that might have made their way into your body and that could cause issues. So we're going to be focusing on these sprinkles on the surface of donuts. And these sprinkles, like I said, were the antigens that are on the surface of the red blood cells. So some people have donuts where they have A sprinkles. So they have A antigens on top of their donuts or on top of their red blood cells. We call these individuals type A blood individuals. Some individuals have on their donut B sprinkles, so they're individuals with the blood type B. Their red blood cells have B antigens on surface. Some individuals are a little bit greedy, so they actually have both kinds of sprinkles because they really love sprinkles. So their donut has A and B sprinkles. So this would be an individual whose blood type is AB, and on their red blood cells surface, they would have antigens, both antigen A and antigen B. And then there's one more blood type. This blood type is blood type O. These individuals have no antigens on the surface of their red blood cells, so they simply have plain old donuts. And I think that this blood type O sometimes confuses people, especially here, because some people end up thinking that there would be O sprinkles. And that's not the case. Actually, in many other countries, we talk about blood type 0 rather than blood type O. So that kind of makes more sense to me, but don't let that throw you off. So there's only antigens A, B, or A and B, or then nothing on the surface of the red blood cells. Well, what we'll see is, which is kind of interesting, that this is the very basis of the ABO phenotypes, so the appearance of the red blood cells. And remember this, in this analog, the sprinkles were the antigens on the surface of the red blood cell. But we'll find that those individuals with type A had those antigen A's, type B had antigen B's, type AB had both kinds of antigens, antigen A and antigen B, and type O had no antigens on the surface of their red blood cells. If we think of the genetics aspect a little bit when we talk about ABO blood system, is that we'll notice is that the alleles for your A, B, and O blood groups are actually coded on the long arm of chromosome number 9. Remember, this is not a Mendelian inheritance pattern. So instead of 2 possible alleles, we're now dealing with 3 possible alleles in total. Let's have a look at those. So what we will use in this coding is as follows. We will use lowercase I to code for a recessive trait. So the trait that we don't see that often, trait that we can only see if both of your parents pass an allele to you for that. So lowercase I codes for these red blood cells that have no antigens for ABO system on their surface. So just the plain donuts. There is that you can also inherit an allele for A antigens on your red blood cell surface. We saw that. And the third possible option that you can inherit from your parent is that you inherit the allele that codes for B antigens on the surface of your red blood cell. So 3 options, but by having these 3 options, we can actually make 4 different kinds of blood groups. Let's have a look at how that works together a little. So the type A blood group is going to have those A sprinkles on the surfaces of their red blood cells. So it's going to be the A antigens on the surface of the red blood cells. The way you can become a blood type A is that you inherit an allele for A blood type, for the A antigens, from both of your parents. So both your mother and father give you the allele that signifies that, oh, this individual is going to have blood type A because they're going to have A antigens on the surface of their red blood cell. The other way how you can become a blood type A is that one of your parents passes you the allele that makes you a blood group, makes you have these A antigens on the surface of your red blood cell, and the other parent just gives you the plain old donuts, so they give you the recessive allele. Remember, recessive allele always got masked if there was a dominant allele presence. And this would be the case in the second example. So this individual would as well appear as a blood type because they have these A antigens on the surface of their cell. Let's have a look at the blood type B. For an individual to be blood type B, their red blood cell surface contains B antigens on their surface. So remember those B sprinkles on donuts. The way you can become a blood type B is that both of your parents pass each one an allele that makes you have B antigens on your red blood cell surface, or only one of your parents passes that allele for B antigens and other parent gives you a recessive allele. And again, recessive allele would get masked and you would end up with the B antigens only on the surface of your red blood cell. There's only one way how you can become a blood type AB. And the way how you become a blood type AB is that one of your parents passes you an allele for A antigens and the other one passes you an allele for B antigens. And that way, your red blood cells will have on the surface of them both A and B antigens. And finally, if you're blood type O, remember there's no O sprinkles. Instead, like I said, I would prefer to call it blood type 0, but we're not there yet. Unfortunately, changing the world. So blood type O has no antigens on the surface of their red blood cells. So they have inherited recessive allele for their blood type from both parents. So both parents must have donated this each one recessive allele for you to be blood type O. I hope that makes sense. To really understand the transfusions that we're talking about, donating and receiving blood, we have to talk about antibodies. So far, we have talked about antigens, but let's talk about antibodies a little. So antigens were these A and B sprinkles on the red blood cell surface, so they help to identify cells with each other. So self-antigens tell the body that it is you, it's part of your cell system. But if there is a foreign antigen on the cell surface, your body is able to tell that, oh, this is a stranger. And if antigens that are not yours are found, your body is designed to remove or kill these cells. And this happens with antibodies. So if you've never seen an antibody before, here you can see it. It kind of is a little bit like this lock and key mechanism that we've seen before. This is just a schematic diagram of what an antibody would look like. An antigen binds to the end of it. If you're exposed to antigens that are foreign to you, you will make antibodies against them. So this is a reaction, and this reaction is something that we actually will talk about as agglutination. So if I'm really cutting some corners straight here, you can consider agglutination as red blood cells kind of clumping together, and that would be a bad. So to understand the donor and recipient matches, it's important that no foreign antigens are allowed. So how we're going to work is that we're going to mix 4 different blood types and see what happens, and most importantly, why it happens, what we'll end up seeing. So here we have normal blood smear. This is what your blood should normally look. You can see a lot of red blood cells, these nice rounded flattened discs evenly spaced apart. If you have antigens and antibodies that are reacting against each other, so here we have foreign antigens that have entered the body with the red blood cells from a donor, and your antibodies are now attacking these foreign antigens, what will happen is known as an agglutination. So in agglutination, these red blood cells end up clumping together. And that's not good. That's going to cause us a world of trouble. And let's have a look of now how we can end up in these different scenarios of trouble. So again, we are back to these tables that we saw before, and rather than memorizing, we're going to make our own. And we're going to look at an individual whose blood type is A. That's what we're going to start with. So the phenotype was always the visible, observable characteristic. So this is the blood type A. An individual whose blood type A will have on the surface of their red blood cells antigens for A. They will have A antigens on their surface. So remember those donuts with A sprinkles on top of them. If you are an individual who has blood type A, so your red blood cell has A antigens on its surface, your blood plasma will include antibodies for anti-B. So you're constantly looking if there is a cell that would advertise that, hey, I have B antigens on my surface. And if your body were to find that kind of a cell, it would be able to say that, hey, this is not part of me. This is not part of the host. This is a problem and we need to deal with it. So that's kind of a foolproof, safe mechanism that we built in. So let's have a look of what can happen. We mix for a patient whose blood type A. If we give them blood from another individual whose blood type is A, both of these red blood cells will have those A sprinkles on top of them. So they both will have A antigens on their surface. Your body will naturally have in the plasma antibodies for anti-B. Well, there's no anti-B from the donor who gives you that blood, so we're good. The blood looks the way it should look. There's no adverse reaction with this kind of a transfusion. Well, for an individual whose blood type A, if we then give that individual blood from another individual who is blood type B, we're going to have a world of trouble. This is because, remember, your normal red blood cells, if you were blood type A, contained on surface of the red blood cells antigens for A antigens. And your normal plasma contained antibodies for anti-B. So your body is constantly looking, is there a cell around that would advertise that, hey, I'm a B kind of a sprinkled donut. And if it finds a cell that advertises that, yes, I have B antigens on my surface, the antibodies in your body will detect that this is an invader. This is not part of us. This is not who we should be. And that's exactly what happens when the donor of the blood type B donates blood to a recipient who's blood type A. So it's the B antigens on the surface of the donor's blood that cause a reaction with the antibodies, anti-B, of the recipient's plasma. An agglutination will happen. You cannot mix these two. Bad. Well, same happens if you are blood type A and you receive blood from another individual who's actually blood type AB. So let's think of that a little bit. The A part of the donor would be fine. That's not an issue, having those A antigens on the donor's red blood cell surface. Because after all, you had A antigens on the surface of your red blood cells. So that's fine. But it is this B portion. Remember, this individual with blood type AB had both antigen A and antigen B on the surface of their cells. So because they also have these B antigens on the surface of their cell, well, your body is constantly looking with the antibodies for anti-B, any cells that might have B antigens on their surface. And if they find, they say that no, this is not part of me. This is an invader. This is a foreign thing in the body. We do not want that happening. And there will be an adverse reaction, agglutination of these two blood types. So not a good match. But let's think of the last possible case. In this case, again, we are blood type A. We will have on surface of our red blood cells those A antigens. Our plasma will contain antibodies for anti-B. And if you receive blood from a blood type O, well, their red blood cells have nothing on their surface. They have no antigens, so they're not going to be a problem. We're like, okay, this is probably okay with us. Nothing for your antibodies to react against. So if you're blood type A, you can totally receive blood from a donor who's blood type O or who's a blood type A, which is a match to yours as well. But as soon as we see that B, that's going to be a problem. Let's look at the next blood type. This blood type B carries on the surface of its red blood cells B antigens. Its plasma is going to carry antibodies for anti-A. So screen anything that has an antigen A on its surface is a foreign and is a potential threat, and we have to deal with it. So therefore, if you donate blood to an individual whose blood type A and your blood type is, whose blood type B and your blood type is A, well, now the recipient's blood and the plasma of the recipients where the antibodies for anti-A are going to react with your A antigens on the surface of your red blood cells. So there's going to be an agglutination reaction. Donating blood for an individual who's blood type B, when your blood type B as well, is not going to be a problem. Both of you will have antigen Bs on the surface of your red blood cells. Nothing to worry, because there's not going to be any A antigens on the surface, so those anti-A antibodies are going to be just fine. If you are donating blood to an individual who's blood type B and you happen to be blood type AB, that would be a problem, even though the B part would be okay, because your red blood cells contain both A antigen and B antigen. The B antigen part of that was okay because the recipient is going to have the same as well on their red blood cells. But you also introduce now antigen A's on the surface of your red blood cells. Well, this is going to be a problem because the recipient's antibodies in the plasma, anti-A antibodies, are constantly looking for something with an A antigen. They see it on your blood and they will mark your blood as a problem that we need to deal with and attack that. So there's going to be an agglutination reaction, negative reaction taking place. Finally, let's think of if you are blood type O and you donate your blood and it goes to a recipient who's blood type B. The blood type B individual is going to have those B antigens on its surface, red blood cell surface. Its antibodies for anti-A are screening around for anything with an antigen A on its surface. Well, if you're blood type O, your red blood cells have nothing on their surface. You're good. We can give your blood to this individual who's blood type B as well. Let's have a look at the blood type where you are blood type AB. So now your red blood cell surfaces will naturally have both antigen A and antigen B. Therefore, your plasma will not contain antibodies for either one of those, because we're not going to want to attack your own body. Therefore, if you receive blood from individual A, there's no problem because there's nothing to attack the red blood cells from the individual with blood type A. If you receive blood from individual with blood type B, again, it's not going to be a problem because we have no antibodies to attack anything. So that's a good match. Individual whose blood type AB can also donate their blood to another AB individual. They're the same, the red blood cells, so there's no issue. An individual whose blood type O, again, those are the plain red blood cells, no antigens on the surface, so there's not going to be any reaction against those either. The final one that we have left is the blood type O. If you have a blood type O, remember your red blood cells now have no antigens on its surface. So you have those plain donuts. However, your plasma will contain antibodies for anti-A and anti-B. So we're constantly looking if there's any cell that carries antigens for A or B on their surface, they're a problem. So receiving blood from a donor who's blood type A, if you are blood type B as a recipient, that would be a problem because your own natural antibodies for anti-A and anti-B, the anti-A's will attack now the donor A who's blood type A, their red blood cells. Same would happen with blood type B donor because your natural anti-A and anti-B, the anti-B part antibodies will now attack the red blood cells of a donor B because donor B's blood carries now on the surface of the red blood cell those B antigens. If the donor is AB and you are blood type O, well, again, your blood contains naturally antibodies for anti-A and anti-B. It's going to attack those on the blood that you received from AB individual on the red blood cells. And finally, if you receive blood from a donor who's blood type O, there's nothing to attack. So the anti-A and anti-B, they have nothing to attack. You can receive blood from another O individual. This table actually is rather convenient. It tells us something else that's going to be rather interesting. It shows us that anyone who's blood type O can donate blood to anyone. And anyone whose blood type AB can receive blood from anyone. Therefore, we talk about individuals with blood type O as universal donors, and we talk about individuals with blood type AB as universal recipients. So the big message here was that you cannot give foreign antigens to someone. That would be a problem. And now that we've discussed about blood type A and blood type B and blood type AB and finally blood type O, I think we're ready to have a look of the RH groups. And it turns out that there's actually 52 different kinds of RH agglutinogens or RH factors, if we wish to call them that. The most common ones are C, D, and E. And in fact, if someone is Rh positive, they're gonna have antigen D on their red blood cell surface. And this actually covers 85% of Americans. So most of Americans are Rh positive. They will have antigen D on the surface of their red blood cells. To become Rh negative with anti-Rh antibodies, which would be a bad thing, it's needed that there's a pregnancy and mixing of blood from the fetus and mother, and then the second that causes the antibodies to be produced, and second pregnancy would be problematic. Or that you receive a blood transfusion first time, it's not a problem, but second time if you receive again this non-match, there would be problems. And that really brings to the conclusion what I was going to say about the kind of preparing you for this lab. So that's the theory, or that's the logic behind everything that we're having a look. So now let's jump on and have a look of our lab and our lab worksheet a little bit together. So what we're going to talk about is the questions that you will see on the lab And we will see that you really could complete the questions 1 to 4, even if you did not attend to the lab. But it's the question 5 where you had to be on the lab, you had to complete one of these blood testing cards, and I'm expecting to see either a drawing of that or a picture of that on your lab report if you're looking for points for the question 5. So let's have a look at what we can find. I'm not going to go through the theory again. We've had a look of that together. What I'm going to do instead is show this picture. You remember on this lab, we collected 3 blood samples from you, one to inside each of these pairs, or inside one of each one of these pairs of circles that we saw. That's where the blood drop went. And then we put the second circle right next to it, our anti-serum. So we put the anti-serum on the circle next to it. And then we mixed up the two. And as you're mixing them up, what happens to that mixture is the part that's going to be important. So what we can see on this PowerPoint, on the first pair, is that underneath the blue bottle, which contains anti-A serum, the blood has clotted. So there's been an agglutination reaction. You can see those clotted red blood cells. For anti-B serum, when it's been mixed with the blood, they've just made a kind of nice mixture. There's no clots visible there. So now you can do that for all of the different combinations for anti-A serum, anti-B serum, and for anti-D serum, which was the factor for Rh positivity or negativity. I'm not showing that on this particular picture. We're just sticking with the AB blood types here. So now this individual whose blood will have an agglutination reaction to anti-A serum, but not to anti-B serum. What we find is that the red blood cells will contain A antigens, but not B antigens. So therefore, there will be blood type A. An individual who will have no reaction of their blood and anti-A serum, but will have a reaction for the blood and anti-B serum, they will be blood type B because on the surface of their red blood cells, naturally, there will be those B antigens and only those. Now we have an individual who has a reaction for both anti-A and anti-B serums. So there's agglutination for both. So their red blood cells must carry antigens for both A and B, so they're going to be blood type AB. And finally, individual who, when you mix their blood with anti-A serum, there's nothing that happens. You mix their blood with anti-B serum, there's nothing that happens. So therefore, there's no antigens on the surface of their red blood cells, and they will be blood type O. So if you did this and you found out what ABO blood type you were, you're halfway there. The only remaining question that we have is that, are you going to be Rh positive or are you going to be Rh negative? And the way we're going to find that out is by looking whether you have D antigens on the surface of your red blood cells. So let's write a note about that. We're considering whether you have D antigens. And you remember, if you had D antigens, that meant that you were Rh positive. So therefore, if your blood formed this agglutination reaction with anti-D serum, it meant that you were Rh positive. And this was the case for 85% of Americans. So if you're in that group, you're in a good group. You're amongst a lot of the peers. So now that you did those 3 tests, that allowed you to define whether you were blood type A, B, AB, or O, and whether you were Rh positive or negative. So that was the big thing that I wanted to do with you as your kind of hands-on lab part. What I want to do next is to have a look of a little bit of these combinations of questions that I had. I asked from you on the worksheet that, okay, I'm giving you your blood type. You have to then tell me what kind of antigens you have on the surface of your red blood cells. So what kind of sprinkles do you have on your donuts? And then finally, we will look at what antibodies are carried in your plasma. So what are you screening against all the time and worrying that if you find that that must be an invader? So let's have a look of that together. So first of all, if you're blood type A positive, that means that you're going to have antigens for A on the surface of the red blood cell, and you will have antigens for D, because remember, if you had Ds, that meant D antigens on the surface of your red blood cells, that meant that you were Rh positive. So therefore, your plasma will carry antibodies for anti-B, and if there's any red blood cell that carries B antigens on its surface, we know that that's an invader and there needs to be an agglutination reaction against them. Same logic can be used to work out the blood type A negative. So there's only going to be A antigens because negativity meant that there were no D antigens. So the antibodies in the plasma are going to be anti-B and anti-D. For blood type B positive, you're going to have B antigens, and you're also going to have those D antigens around. And we're constantly screening with the antibodies for anti-A, whether there's going to be any antigen A's around. And if there are, those cells are definitely invaders, pathogens to your body. If you're blood type B negative, you're going to have only B antigens on the surface of your red blood cell. Remember, negativity meant that there were no D on as antigen on your red blood cell surface. So your plasma will, in this case, include antibodies for anti-A and anti-D. If you're blood type AB positive, you have pretty much everything going on in terms of the antigens on your red blood cell surface. So you will have antigen A's, you will have antigen B's, and you will also have antigen D's, because remember, that gave you the positivity for Rh marker. Your antibodies in plasma are not going to be any of these. So there might be other, but we're now focusing on the AB, O, and Rh. So there's nothing. AB negative, we're going to have both A and B antigens. We're not having D antigens. So therefore, the antibodies in the plasma are going to be those antibodies for anti-D. Blood type O positive, well, there will not be A, there will not be B, but there will be the D that gave the positivity. If you had no A and B, that meant that you were blood type O. So therefore, your antibodies in plasma are going to be anti-A and anti-B. If you find any red blood cells that have A or B antigens, they are going to be invaders. They're going to be a problem for us. If you're O negative, you will have none of these antigens that we've discussed on the surface of your red blood cells, but your plasma will carry anti-A, anti-B, and anti-D antibodies. So I hope that that makes sense. I offered a few other challenging questions about what blood type would agglutinate with anti-A, anti-B, and anti-D serums. Well, that would be AB positive. What blood types would agglutinate with only anti-A serum? Well, that would be A negative. Let's talk about what blood type would agglutinate with anti-B and anti-D serums. That would be B positive. And I'm asking what blood type would not agglutinate with any antibody serums, and that would be, of course, the O negative. Next question asks you what blood type would agglutinate with only anti-D serum? That would be O positive. The next question asks what blood type contains no anti-A antibodies? That would be either B or O blood type. In terms of what blood types contain no antigens at all, that would be O negative. In terms of what blood types would agglutinate with anti-B serum, that could be either B or AB blood type. We don't have enough information to say. What blood types could agglutinate with anti-RH serum? And that would be any Rh positive blood type. And then I'm finally asking what blood types could agglutinate with anti-A serum? Well, that's going to be A or AB blood type. So that should be clear from what we did, looking at those blood bottles, looking at what sort of mixtures we could do, as we saw in these pictures. Now I think I'm ready. I think we're ready to have a look of the blood transfusion. So mixing two types of bloods. Let's have a look of that a little bit. And that was the question 3 on the lab worksheet. And we're going to start by talking about which blood type is the universal RB red blood cell donor. And that was, of course, our individuals with a blood type O negative. I was asking from you also that could a person with type A positive receive type A negative blood? And this would be fine because the ABO blood groups match and the Rh positive can receive Rh negative blood. So that's not going to be a problem at all. That's going to work out just fine. Let's have a look at the next scenario where we're asking could the person with type O negative blood receive type B negative blood? And this would not be possible because the ABO blood groups do not match and would cause the transfusion reaction, so that agglutination that we saw, and also because the O negative plasma would agglutinate with the B antigens on the B negative's red blood cells. Let's have a look of this mixture where a person with type AB negative, could they receive blood from type A negative person? Yes, they could. ABO groups and Rh factors match. So that's a good thing. Since AB red blood cells have both A and B antigens, the body will not attack red blood cells from type A blood with only A antigens. So that's okay. Final question is a little trickier. Let's consider this. So could a person with type B negative blood receive a type O positive blood? So we need to do a little bit of thinking here. Yes, this is possible, but this is possible only for one time because the ABO blood groups do match. Since type O has no ABO antigens, the B plasma will not agglutinate with that. That's fine and all good. But where it gets tricky is that Rh negative patients can receive Rh positive blood, but only one time, as this will now trigger the development of the anti-D antibodies, and this would cause agglutination of any subsequent transfusion with Rh positive blood. So this is the same as we saw when we talked about, for example, the mother and a child and first pregnancy. being okay, but second pregnancy, if there's been mixing of the bloods during the first pregnancy, being problem. The final one that I'm kind of curious to discuss a little bit with you has to do with the plasma transfusions. So which blood type is the universal plasma donor? That would be our AB positive. And we're looking at the person with type A positive blood receive type A negative plasma. And yes, this would be okay if the donor did not have an exposure to antigens, D antigens. They would not have anti-D antibodies. I'm asking about, could the person with type O negative blood receive type B negative plasma? And here, yes, the ABO groups and RH factors match and would not cause a transfusion reaction, would not cause that agglutination reaction to take place. Let's have a look of the next scenario. In this scenario, we're asking that could a person with type AB negative blood receive type A negative plasma? And this would not be possible because the anti-B antibodies from the A negative plasma would agglutinate with the B antigen on the AB red blood cells. And final one that I have here is that could a person with type B negative blood receive type O positive plasma? Well, this is not possible because the anti-B antibodies from the O positive plasma would agglutinate with the B antigen on the B red blood cells. So that was quite a bit to get our head around to, but a really, really useful exercise. I will be honest, I think it's useful that you learned this. It's really important that you learned this. I probably will not ask about that again in the exam, but I think it's important that you have a handle of what's happening here. And you remember that the final question, the question 5, was all to do about you doing your own blood typing. So if you missed the lab, these are the points that you will miss unless you made alternative arrangements with me ahead of the time. Remember to submit your lab on Canvas in an electronic format by the end of this week. Remember to include not only the lab worksheet, but also the picture or a drawing of your blood typing card. And you need to also analyze that. Tell me what blood type you were and why. So explain that, remember that. And that really completes everything that I had planned to cover during today's evening review session. It's important for me that these are useful for you. So if you join and you have particular topics that you're interested, I will always facilitate those. So that's why these sessions may take many different shapes, many different forms, depending on who's attending and what kind of questions you have. So that's going to conclude our evening session. I hope it was helpful for you, and I look forward to seeing you next week. Remember, in the meantime, all you have to do is drop me a message on the Canvas, and I definitely will be here to help you every step of the way so you excel in the exam the way that, or in the coursework and on the course the way you should. Have a good rest of the night and have a very good weekend until I see you next week again.